Alright guys, today we're going to learn a little bit about the other types of inheritance. We've already covered dominant and recessive, sex-linked, blood type, which is a multiple alleles, autosomal dominant diseases, and autosomal recessive diseases. So now we're going to go into other areas of incomplete dominance is another type. Codominance is again the blood type, specifically AB blood type, where they're both being revealed, and also polygenic inheritance, Pleiotrophy, and environmental influences. So let's start off with incomplete dominance. Incomplete dominance is also known as intermediate inheritance. So sometimes people use incomplete or intermediate. So either one of those would be the same thing. And that's when the phenotype is something like in the middle between either extremes. So if we think about our prefixes, inter means what? A little practice here for our prefixes. And that means between. Inter is between. So it almost looks like you've blended both parents. So if I had a flower that was red and I mated it with a flower that's white, if it was inherited by an intermediate pattern or an incomplete pattern, we would end up with a pink flower. So we need to figure out a way to actually write this differently than we've written other things. So how we go about that is in intermediate or incomplete dominance, we would write the color with C, and then we actually put superscript colors up here. So this would be CB. CB would give us a black chicken. CB/CW would give us something in between, and I know you're saying black and white would give us gray, but they typically call this chicken a blue chicken, so I apologize for that it's not gray, but it's something in between either extremes. The white would be CW/CW, so the only way we get a white chicken is if we have these two alleles. The only way we get a black chicken is with this, and if we get something in the middle, it would be this. So I know that you're dying to see this, but this is a picture of that particular chicken. And so this is an example of a blue chicken, and that is something that's in between both of its parents. Here's our Punnett square for another flower. It's actually called a snapdragon that is inherited from an intermediate inheritance pattern as well. CR, the R would be red, so this is a red flower at the top, CR here, and that's mated with a white flower, CW/CW. And when we cross that, we see that all of the offspring are CR/CW, and those would be actually pink flowers. So the way we would figure out the intermediate inheritance, because it's kind of like, well, how would I know what kind of inheritance pattern? Well, if you look at the parents and the offspring doesn't look anything like either one, it is something in between, we would know that it would be an intermediate or incomplete dominant. So here all of the offspring would be pink. And if we were to look at something like this, is our white flower, so this would be CW/CW. This is our red flower, CR/CR. Here's all the pink flowers that it makes. So this is the parental generation. This is the F1, so this is filial 1 generation. So if I took this pink flower and I mated it with a white flower, so this would be CR/CW mated with a CW/CW, we would see that 50% would be pink and 50% would be white. But what's interesting is if you took two pink flowers, again this is CR/CW, CR/CW, and if you would actually do a Punnett square with that, 25% of those flowers would be white, 25 would be red, and then we'd have 50% would be pink. So this is an interesting scenario, is that we can have white and red flowers for grandparents. They would make all pink, but then if these two pink flowers were to mate, they have those alleles in there. They have a red allele and a white allele. So that has a possibility of coming out in that next generation, which we call the F2 generation or filial 2 generation. So this is a typical what we would see in that situation. So incomplete or intermediate inheritance, again, is some form of, looks sort of like a blending inheritance. opposed to codominance, co meaning together, like co-ed, jointly mutual. Both alleles are expressed together, and again, we've already done blood type. So a classic example of that is AB blood type, where the person that has AB blood type makes A surface antigens, and specifically their glycoproteins on their red blood cells that are like tags for that red blood cell. And they also make surface antigens that are B surface antigens or glycoproteins. So when we have codominance, if I had a red and a white, we would actually see both of those. So we see red and white. It's not blending, we see both distinct characteristics. So a picture of this would be something like this rhododendron, which we see some red and we see some white. It's not pink, but we see both colors distinctly. So this is a nice summary slide of different ways. We already know dominant and recessive. My classic focus here is to really look at those heterozygous. So if I have this heterozygous, this big T, little t, and if we said that big T was tall and little t was small or short, this person would look tall, right? In codominance, it would be a funny thing. We would see one and we would see the other. So in the heterozygous here, we see both alleles. We see both of them together. In incomplete dominance, we don't see either parent. We see something that's in between. Again, if we have red and white, we would have pink. So here we want to focus in on the heterozygous. So the heterozygous here looks like the dominant. Heterozygous here shows both traits. In incomplete or intermediate inheritance, we see something in between. Okay, so it's something in between the homozygous dominant and the homozygous recessive. Another type of inheritance is polygenic. Poly means, figure this out, and actually I've kind of put two, I put a root here and then a suffix. So IC means pertaining to, and what does gen mean? So to see if you can kind of practice your prefixes here. But the definition as a polygenic trait is that when one or two, actually two or more, genes affect a single character. A classic example of that would be height. Okay, so when we took, say this was our class, and this is the bell-shaped curve of everybody in our class, most of the people would be in the middle, let's say for height, and then there'd be some extremes. Some people would be really short, like Miss Oz, and some people would be really tall, like say Scott, or some other students. So we have the vertically challenged people over here getting to our average height people, and then going down here to our tall people. So polygenic inheritance really always, anytime you see a bell-shaped curve, you know that you've got some form of polygenic inheritance, because not everybody is going to be here, not everybody's going to be here. We're going to follow some kind of pattern here. So height, skin color, hair color, there's so many genes, and so they're trying to show us with these that there's 6 possible genes, 6 possible alleles here, genes here, that will contribute to a person's height, hair color, or skin color. Another classic one is eye color, and this slide always freaks people out, but I love to show this because here clearly we have a bunch of blue-eyed people, but their blue eyes are totally different. This person has white rays and a darker blue background. This person has a little bit more of a faint blue background here and white rays, but you can see how much lighter their blue eyes are here. This person has that golden color, almost a reddish color in the center, and so there's many genes that actually contribute to a person's eye color. It's not just like a person has brown, blue, and green. There's all these different factors that will affect eye color, and so that's a classic example of a polygenic inheritance. Pleiotrophy is when almost the opposite to polygenic. Polygenic is polygenes, many genes contributing to one trait. Pleiotrophy is when one gene affects so many traits. So sickle cell disease is a classic example of this. So it's one gene on a chromosome, But that one little gene, if you have that gene for sickle, you're going to have so many problems, and that's going to be manifested as kidney failure, heart failure, brain damage, and some other things here. In the book, they use an example of the SRY gene that codes for a protein to activate other genes, so that has many, many effects. This slide kind of summarizes how one gene, so one gene for the sickle cell allele, which affects hemoglobin, which makes our red blood cells look like this. If our red blood cells are sickled, then that leads to red blood cell breakdown, clogging of blood vessels, accumulation of the spleen, all these different physical manifestations. So you could have brain problems, heart problems, paralysis, so all these different things just because of one gene, and that would be an example of pleiotrophy. The environment can also affect how our genes are expressed. So in the Himalayan rabbit, if you were to look at a Himalayan rabbit, where they're warm, their fur is really, really pale, and where it's colder, they have black fur. So that's the environment contributing to this. If I were to be born in Brazil, I'm really fair, but maybe I would have darker skin color. So, you know, but if I lived in Greenland, I might be even more fair than I am now because I'm not being exposed to as much sunlight. The other thing is, even if you're genetically predisposed to be really tall, like I could have been 5'8", say, but maybe I didn't meet my calcium needs, I didn't meet my protein needs, and if I didn't meet them, that's my environment. Maybe I was malnourished, maybe my parents didn't have enough money to give me food, so that my environment contributed to the fact that I would be short. The book also gives an example of environmental influences that can affect phenotype. So I'd like you to go to your book toward the end and actually look through that and see if you can bring to class another example of environmental influences and how that can affect someone's phenotype. One last example is this particular flower, which is a hydrangea. This is a classic New England flower, and that if you plant this particular flower in different soil pHs, you get a different color flower. If the soil pH is high, you get a pink flower, but if you acidify the soil, you can get this nice blue hydrangea, and that's what everybody tries to get to. So they get these nice blue, purpley blue flowers. So those are some examples of how the environment influences our genes. So make sure you go back and look at some other examples in the textbook and bring those to class so that we can discuss them next time. Have a good night.